Welcome to the first Skope webinar. This webinar will mark the arrival of the Skope webinar series, in which Skope users and employees will introduce field monitoring from a variety of viewpoints. My name is Jasmine, and this is Zoltan Nagy. He's the head of MRI Physics at the University of Zurich. And today he will present two examples how he used field monitoring for his research in neuroscience.
So let's start. As Jasmine said, I will present two examples of how we use field monitoring in our lab, or what kind of projects field monitoring enabled us to do. I work at the University of Zurich at the SNS Lab. It's a laboratory for social and neural systems research. I have a disclaimer. I do this presentation now as a customer. I'm a customer of Skope. I went through all the steps of applying for grants, coming up with an idea and a project, or a series of projects, applied for a grant, got the grant, and then I put in an order for the field monitoring equipment from Skope. So that was years before I started working for Skope. But now I work for them part-time, so you should know that. But anyway, today I'm a customer, so I will speak as such. Because you indicated that you don't have any, or enough of you indicated that you would like to know about field monitoring, I give you a few slides of intro. So field monitoring is an NMR technique. So you make an NMR measurement just like you do with the actual MRI scanner, and the probes, the field monitoring probes, are seemingly simple. You have a drop of liquid. It could be different materials in a tube, and you have a coil around it, so you can excite that liquid and you can receive the signal from this liquid. So this is the schematics on the left, and here you see an actual implementation on the right. This tube is then filled up with some other material, so that its susceptibility balances the neighborhood, so you have a nice homogeneous field throughout the droplets, so you have a long lifetime of the FID that you measure from it. I hope that's clear. And if you would like to see more, you can look in this paper that I took this from. The actual implementation that Skope sells, and the one that we bought, they call it the clip-on system. So the probes that you saw on the previous slide are housed in these little black rectangles, and it's a clip-on system in the sense that you clip it to whatever head coil you want to clip it to. So whatever you use for your measurements, it's a flexible installation. You can attach these probes to your regular setup. There are 16 probes, so you can fit up to 16 terms, so up to third order. In the back here, behind the head coil, you see what is called an RF box, RF front end. So I'm a customer, so as a customer, I don't know much more about it. I will not say much more about it. But anyway, that's one of the components still of the setup. And outside of the scanner room, you will have a spectrometer. So this handles the transmit and the receive, as well as the data storage. And then from the data that you measure, all the spherical harmonic fitting and display is also done with this acquisition system. And the setup is something like this. You put the probes around your coil, and then the probe is attached to the TR box that I showed you in the previous image, and then you connect it to the inside of the Faraday cage. That's the dotted line around the scanner room. And outside the scanner room, you have the acquisition system where you actually run the experiment from. So I hope that was enough of an introduction to start with. If you have more questions, we can get into it in the Q&A session at the end. Okay, so now we come to my actual presentation. I will talk about cortical parcellation with high angular resolution diffusion imaging data. That's what I would like to do in my lab. And I will talk about the limitations that we ran into previously. Also, I talk about another application, which is measuring and correcting gradient nonlinearities in diffusion imaging and the limitations thereof. Then I will show you our field monitoring setup at the SNS Lab here in Zurich, how we installed those clip-on system. And and I'll show you our current data quality, and finally I'll finish off with what we would like to do with the data that we have. So first, cortical parcellation. Just excuse me, I have to take a cup of water. Sorry. So I would talk about the first application. We published the paper after 5 years of trying. It was not an easy project. And this is using this Hardy data to try to discriminate cortical regions. So our hypothesis was that the diffusion signal in a voxel that contains gray matter, cortical gray matter, should be useful for fingerprinting that tissue. The idea came from an earlier abstract from Derek Jones and Sean Dione, where they, rather than fitting the diffusion signal evolution over the number of diffusion directions, which is on the x-axis here, and the signal amplitude on the y-axis, rather than fitting this data to some sort of model, like a diffusion tensor, for example, they just said, let's actually look at the time evolution and use this as a fingerprint. And they could use this successfully to parcellate the thalamus, which I guess you know is a cluster of gray matter deep down in the brain. So this was the eureka moment for me. Like, why can't we do this in the cortex? And one of the reasons it's difficult to do it in the cortex is that the cortical tissue is curved and folded. But anyway, the paper that I showed you in the previous slide, that's what it accomplishes, to try to do this idea in the cortex. If we just look at these 4 images, these are the diffusion signal extracted from the cortical gray matter and displayed on the gray-white matter boundary surface. And the four images that you see, 4 subplots that you see here, are 4 different diffusion directions. And if you look at the tip of one of the arrows, let's say this one here on the top left here, you can see at the tip of it that at this diffusion direction you have this red signal. Here it's a much more extended red, here it's much less red, and here it's not red at all. So as you change the diffusion direction, the diffusion signal itself changes, as Derek Jones and Sean Dione showed it in the thalamus. As you change the diffusion direction, you have a large variability. For a long time, this, or I guess still, it's true that the diffusion is considered much more isotropic in cortex, and but what we found was that even though it's isotropic, it's not perfectly spherical. There is some jaggedness to the 3D diffusion profile, and that is very reproducible. Here is just another example. Again, 4 diffusion directions. What I want to show is that just like Derek Jones and Sean Dione extracted data from the thalamus, if you extract it from neighboring voxels in a certain cortical area, you see that the neighboring voxels, the diffusion signal in neighboring voxels is highly correlated. Whereas if you take area 1 and area 2, within each area, although they are correlated, the 2 areas are very distinct. As I said, this is not enough yet, because even if area 1 and area 2 were identical tissue, but one was rotated a certain angle relative to the other, even the identical tissue would show you a different evolution of the diffusion signal, because relative to the diffusion encoding diffusion directions, they will be differently oriented. So you have to deal with that somehow. And I don't want to get into the details, but that's what the paper accomplished that I mentioned earlier. Yeah, that's just to show correlation matrix, the same idea. So the end game of that paper was just a proof of principle idea that if you scan somebody twice and use the first data set to train a support vector machine classifier, and then you classify the second data set, then you can get, I think it was up to close to 90% accurate classification. So there was some signal in the gray matter that you can reproducibly measure. And of course, this is not very useful yet, because you first have to know the regions of interest from which you extracted the data for the training. But nevertheless, it was interesting for us to show, and now based on this, we would like to further develop this technique. The problem is that we run into several limitations because, of course, we use EPI images, echoplanar images for the diffusion readout. So we have resolution issues. The cortex is 2 to 4 millimeters thick, and our data was something around 2 millimeters. So there's a lot of areas where you run into partial volume issues. There are, of course, eddy current distortions, SNR issues, susceptibility distortions, all of which make this procedure very hard and very error prone, and also these steps introduce systematic errors. So, for example, the diffusion signal over time, the undulation that you saw over the 60, let's say 60 or so diffusion directions, could simply be tissue being warped differently in each diffusion direction, and due to the eddy current distortions, you will have a very specific time signature, which is, of course, just a systematic bias. It has nothing to do with the underlying tissue there. So somehow you want to get rid of these eddy currents. And if I just show you some data with eddy currents, just for your eye to get used to it. I don't know how many of you are diffusion experts and how many of you looked at it before, but these videos run through this in more or less the same slice, in the same subject, the 60 or so diffusion directions. And if I don't know what you're viewing it on, but if your display is big enough, and if you can see my mouse, which is over here, I'm looking at the top right now, you can see that tissue is moving in and out of the voxel as the diffusion direction changes. So obviously this would create a fingerprint for you, but it's not very useful. What you see here, the labels are scanner 1, scanner 2, and scanner 3A, scanner 3B. The first three, the top row and the bottom left, are one of Siemens, Philips, and GE each. So all of the scanner manufacturers will do this for you. So eddy currents are present in each case if you do a statistical Tanner encoding. Whereas on the bottom right, you see twice your focus spin echo encoding on one of the manufacturers. Doesn't matter which one. So the eddy current performance is much better in this case on the bottom right. So you don't see tissue moving in and out of the voxels. If you look further, or if you want higher resolution data, then you would see still eddy currents. I mean, the twice your focus spin echo method only corrects for one eddy current time constant and none of the others. So it's not perfect data yet, it's just much more improved relative to the statistical Tanner variant. Okay, I just have to acknowledge that this data that I showed you came out of Siyavush Mohammadi's paper. So if you'd like to look that up, it's over there. So another application that I would like to show you that we also worked on quite a long time ago we started is an idea to use diffusion imaging to measure the gradient nonlinearity of the system that you're working with. It was an ISMM abstract in 2009, so the project was sometimes done before then. I don't know, maybe about 2007, 2008. And the idea is very simple. You put a water bottle in the scanner, and the idea would be that the water diffuses at the same rate in all directions at any spatial location within the water bottle, right? Like, so on the left-hand side, the diffusion shouldn't be higher than on the right-hand side, and so on. And then, so if you do that, in this case, the water bottle was actually a spherical water phantom. You can see that the diffusion constant that you measure has a very specific spatial bias. So in this case, in the axial, on the top left, in the axial, oh, hang on. I think I can turn on my laser pointer trick. Okay, you can see it now. Excuse me, I forgot to do it earlier. So, but in the top left figure, you see that the anterior part is much higher diffusion constant than the posterior. And you might even think that, okay, hang on, this is probably, it may be a temperature bias within the phantom, right? That, I mean, it could be that the front of the phantom is warmer for some reason and the back of the phantom is colder, just by the ambient temperature around there, or maybe there is airflow, whatever. But if you change the diffusion direction, you can look on the right-hand side now, like this diffusion direction 1, diffusion direction 2, then immediately the profile changes. So we inferred that what you measure here is actually the gradient nonlinearity. So where you measure higher diffusion constant, that means the gradient is stronger rather than the diffusion being actually at happening at a higher rate. And if you measure this nonlinearity, you can turn it around and use it to correct a future measurement. So if you know that your b-value here at the tip of the phantom, at the anterior part of the phantom, was not 1000, but it was 1050, then you can use 1050 to calculate your ADC, the apparent diffusion constant. And in the back of the phantom here, where it was only 985, you use that b-value, and then you get a flat diffusion constant. So this project was envisioned and tested and tried in 2009. And we tried to actually make it practical and use it. But what we found was that when you actually want to scan a water phantom with changing diffusion directions, you will run into the same thing as I showed you earlier in vivo data, that you have eddy currents. You can see, I don't know if you see it on your display, but often you have ghosts. I mean, EPI images, we're used to ghosts. And then on the left is the hard data, and on the right-hand side, you see the p-value map, which should be somewhere around 1 in the center. So the nominal b-value, like, let's say 1000 here, and out here at the edges should be dictated by the gradient nonlinearity. But this data, actually, the artifacts are bigger than the effect we're trying to measure. So for a long time, we were bogged down with how to continue. And so meanwhile, other people, for example, Bertram Wilm in Zurich, was presenting data which was looking more like this. If you look at the bottom row, this is, for example, a diffusion weighted image. And this is the mean of all the diffusion weighted images. I think he had 6 in this case. This is a spiral acquisition, a statistical tenor readout. Sorry, statistical tenor preparation and a spiral readout. And then so he could achieve a 34 millisecond echo time on a 3 Tesla scanner, which was phenomenal. And yeah, it's I guess everybody would like to do that if they could. Or at least, even if I can't talk for everyone, I wanted to do this. So based on this sort of hope that I could also do it, we installed our own magnetic field camera. We decided to take these probes that I showed you earlier. But these little rectangles house the probes. They're not all the probes themselves. The probes are in there with some electronics. We decided to install it in an 8-channel head coil, and the way we implemented it was to have this 3D printed insert which holds the probes within itself and the inside of the coil, which you see in part B down here. So you can see here the design from different angles, from the top and from the side. You can see the coil and the 3D printed part. In part C, it's half assembled with the probes inserted, and in part D, you see the entire assembly. And yeah, it's not an easy installation. We had to hire an industrial designer who helped us with this project. But it was very important for us that the installation is pristine, in the sense that we're a neuroscience lab. We cannot have installations that look like a nuclear submarine, and everybody would be scared to go into the scanner. Like, people should not be apprehensive when you do an fMRI study, for example. So this is our final installation. You can see the coil setup with the probes. You can maybe see those little protrusions, which are the probes underneath the insert. Here is the TR box that I mentioned, and you can see the single cable running out towards the back of the scanner. So that's our installation, and we were very happy with it. And as I said, we put a lot of effort into it, because we want to make sure that people that come to our scanner for an neuroscience study go into it as happily as if there was no field monitoring present in these experiments. And so this paper is out. So we would like to know more. A lot of design what's the word? Excuse me. Aspects. A lot of consideration went into designing it, the final installation, the way it ended up, and then you can read about it here if you're interested. So I'd like to show you our current data quality, what we work with now. So this is a diffusion data set in a well-behaving subject. So you see that there is no eddy current issues. If I put my cursor here, you see that tissue doesn't move in and out of the voxel. Every once in a while, you see the image jump. It's not an eddy current and it's not subject movement. It's actually, we're moving to the next slice. So it's several slices and the diffusion directions are up to, I think it's 64. So you see all the 64 directions and then the slices go contiguously one after the other. If that doesn't convince you yet of the data quality, mind you, it's an 8-channel coil, so the data is a little bit noisier than I would like. But this is the mean of all the diffusion data. So we had 64 DWIs, diffusion weighted images, and this is the mean of all of them. And you can see a very nice gray-white matter separation. You can see, because you can see such a clear distinction between the gray and the white matter, you already know that the different diffusion directions were very nicely congruent, very nicely aligned. And if that still doesn't convince you, this is the variance image across the diffusion directions, and you see that there is no edge effect. So you don't see a high variance at the edges where a tissue would be moving in and out of the voxels. So we're very happy with this data quality. We can discuss it later if you see some artifacts in it or see something that you're not happy with. I'd be curious to know. The other project I presented to you was the one with where we tried to use a water bottle, a water phantom, to measure the gradient nonlinearity. This is the data now that we get from the water phantom. So you can see the lack of, or maybe the minimized ghosts and the minimized eddy currents. And on the right is the B-value map. So this is now, as you expect, you have the center where the gradients are linear, and as the diffusion direction changes, that's what the movie is, the different diffusion directions. Then you can see how the gradient nonlinearity changes as you know the X, Y, and Z components of the gradients as they change. So this is our data quality now. This is what we're working with. This is what we arrived at, and our current projects include installing these field probes into a 32-channel head coil. It's a lot of extra work and different kind of work. 32-channel coils are built differently. Then the 8-channel, we need this for SNR. We're working on implementing the spiral sequence so that we can use statistical tenor diffusion encoding and minimize echo time for improving or optimizing our SNR. We're working on the reconstruction pipeline so that people who arrive at our scanner, they can just press start in the scanner and then later just arrive at their data. They don't even have to know that field monitoring was done during their experiment. And of course, all this goes into our main focus, which is what we started with, which is cortical parcellation. And based on that hopefully doesn't have systematic artifacts that would be confounds to our question of how well the diffusion signal fingerprints cortical tissue. I think that's the end of my presentation. I just wanted to acknowledge the help of others. We get support from the Swiss National Science Foundation. When I arrived here, I used to think that I was already an experienced MR physicist, and then you move somewhere else, and then you get humbled by how much you don't know. And I would like to thank Professor Klaus Christmann and his team for all the collaborations that we were able to do together. We learned a lot, and we are very appreciative. Skope, I would like to thank because even before I started working for Skope, some of the pictures I showed you with the industrial designer, the other person in that picture was a Skope employee. So we had a very close collaboration during our installation, and I was very happy about that. So I'd like to thank them. And finally, the PhD student that's working on these projects is Eugene Lee, and and she's doing extremely well, and I just wanted to thank her for all the hard work that she put in. Also, maybe if you're looking for a postdoc, you would be very happy to attract her to your lab some way. I don't know, I'm trying to do the same, so we're in a fight for her. But anyway, she's doing an amazing work, amazingly, and I just wanted to thank her for all the hard work.